Hello everyone, this video is about physiology and ontogeny of immune system, organization and structure of immune system. And this study note is from EPG Pathshala, MHRD, India site. So I would like to express my sincere gratitude to the developing team of this study note. Thank you so much. So let's move on to the topic. Organization of immune system in human. What are lymphoid organs? Structure and function of primary and secondary lymphoid organs. Role of lymphatic system in immune response. The immune system is a complex and highly developed system, yet its mission is simple: to seek and kill invaders. If a person is born with a severe defective immune system, death from infection by virus, bacterium, fungus, or parasite will occur. A lack of immune system cells is also the basis for Discharge syndrome. Improper development of the thymus gland means that T cell production is diminished. Humoral immune response. Humoral immune response: B lymphocyte antigen plus antigen, antibody secreting plasma cell eliminating antigen. Then cellular immune response: dendritic cells, TCR, CD4, TCR, CUB, activated T helper cell, activated cytotoxic T cell, cytotoxic T effector cell, T helper effector cell, cytokines killing the infected cell. Then dendritic cell, activated cytotoxic T cell, activated T helper cell, T helper effector cell, cytokines, cytotoxic T effector cell, killing infected bacteria. Flowchart. The immunological system is comprised of the lymphoid tissues and organs of the body. Lymphoid tissues are widely distributed. They are concentrated in bone marrow, lymph nodes, spleen, liver, thymus, and Peyer's patches scattering in lining of the gastrointestinal tract. The lymphoid system is encompassed by the system of phagocytes. Lymphocytes are the predominant cells, but macrophages, dendritic cells, and plasma cells are also present. Lymphocytes are cells which circulate, alternating between circulatory bloodstream and the lymphatic channels. Organs of immune system. The immune system functions throughout the body. There are, however, certain sites where the cells of the immune system are organized into specific structures. These are classified as primary lymphoid tissues or central lymphoid tissue, bone marrow, thymus, and secondary lymphoid tissues or peripheral lymphoid tissue like lymph nodes, spleen, mucosa associated lymphoid tissue. Blood vessels and lymphatic systems connect these organs and unite them as a whole to function against antigen. Tonsils, thymus gland, see the location, cervical lymph node, tonsils, right lymphatic duct, entrance of thoracic duct into subclavian vein, thoracic duct, axillary lymph node, maxillary plexus, intestinal lymph nodes, spleen, Peyer's patches in intestinal wall, appendix, bone marrow, inguinal lymph nodes. That is organs of the immune system of humans. The next, thymus gland, lobule, medulla, cortex, thymus gland. Then medulla, cortex, capsule, lymph node. Then monocyte, lymphocyte, red bone marrow. Lobule, medulla, cortex, thymus gland. Lymph node, medulla, cortex, capsule. Then lymphatic vessels, spleen, capsule, white pulp, red pulp. Then red bone marrow, monocyte, lymphocyte. Primary lymphoid organs. Primary lymphoid organs are also called as central lymphoid organs. Primary lymphoid organs are also called as central lymphoid organs. These are responsible for synthesis, and maturation of immune competent cells like bone marrow and thymus. All cells of immune system are formed in the bone marrow. During hematopoiesis, the process of formation of blood cells, bone marrow stem cells develop into either mature cells or precursors of cells that migrate out of the bone marrow to continue their maturation elsewhere. The bone marrow produces lymphocytes, B cells, T cells, and natural killer cells, granulocytes like eosinophils, basophil, neutrophil, monocyte, and dendritic cell, in addition to red blood cells and platelets. So all cells of the immune system are formed in the bone marrow. All cells of the immune system are formed in the bone marrow. During hematopoiesis, the process of formation of blood cells, bone marrow stem cells develop into their either mature cells or precursors of cells that migrate out of the bone marrow to continue their maturation elsewhere. The bone marrow produces lymphocytes, B cells, T cells, and natural killer cells, granulocytes, eosinophils, basophil, neutrophil, monocyte, and dendritic cells, in addition to red blood cell and platelets. Bone marrow is not a disorderly mass of packed cells. It has a structure and system of its own. It may, if any long bone is cut half lengthwise, distinct changes in color can be noticed. Some areas would be very red, and these areas should be the site of RBC's production as well as white blood cell production. Other patches would be yellow in color. These yellow colored patches are as a result of adipose fat cell accumulation. The yellow areas are relatively inactive in healthy people, but when an individual's blood system is stressed in some way, these yellow patches convert into blood cell forming tissue. So yellow region are the emergency standby cells. Here we could see blood vessels. This is the structure of the bone showing bone marrow. Blood vessels are there. Marrow we can see. Then peritoneum, membrane covering bone, or periosteum actually. Cortical hard core, trabecular spongy bone, articular cartilage, epiphyseal plate, marrow and medullary cavity. Bone marrow cells need a scaffolding to help them organize and be systematic in the production and release of cells into the bloodstream. The scaffold is provided through three main structures: the blood capillary network versus sinus and a reticular fiber framework. In many bones, the capillary network originates from a single artery which enters the bone close to its center. Inside the bone, it divides and continues to subdivide along the length of the bone. Small capillary branches radiate off the larger blood vessels to reach the extremities of the bone marrow. These capillaries connect up with a second system of vessels called the venous sinus. Sinus is Latin for cavity. The connection between venous sinus and blood capillaries is continuous. So the scaffold is provided through three main structures: the blood capillary network, venous sinus, and a reticular fiber framework. In many bones, the capillary network originates from a single artery which enters the bone close to its center. Inside the bone, it divides and continues to subdivide along the length of the bone. Small capillary branches radiate off the larger blood vessels to reach the extremities of the bone marrow. These capillaries connect up with a second system of vessels called the venous sinus. Sinus is Latin for cavity. The connection between venous sinus and blood capillaries is continuous. Blood from the sinuses is collected by venules and veins which merge together to leave the bone via a single vein. So the blood system of a bone is closed, one entry and one exit. Leukocyte, fat. Venous sinus have some exceptional properties that other blood capillaries or arteries veins don't have. Venous sinus are the vessels into which blood cells are released after being produced from the marrow cells. These sinus are barrel shaped and expand and contract to accommodate the variable production and release rate of blood cells. The sinus either have perforated walls or don't have walls at all. Blood cells that are ready to leave the bone migrate to these sinuses and release themselves into the sinus. If there is a vessel wall present, the new blood cell find a perforation and squeeze themselves through the opening. And usually the hole is too small for easy passage of cells, so blood cells have to force their way through. The small hole size ensures blood cells already in the vessel don't readily get back into the bone marrow. Finally, we have a reticular cell and fiber meshwork which forms the underlying scaffold for supporting the bone marrow cells and helping keep the blood capillaries in position. This reticular framework basically stops the marrow cells from sloshing over around and getting damaged when we move. General changes in the color of the red marrow can sometimes be observed if an individual is under challenge from a chronic challenge. The red marrow changes to pale pink. The marrow switches from producing red blood cells to concentrating on making white blood cells. This may result in anemia as a side effect of hepatic infection. Thymus. The thymus, in evolutionary terms, is the derivation of fish gills. The thymus is believed to produce lymphocytes which colonize the lymphoid tissue. This view is supported by several facts. Removal of thymus from newborn mice leads to the development of fatal disease. Congenital absence of thymus or thymus aplasia, incomplete or defective development of the thymus, results in immunologic deficiencies. In the adults, removal of the thymus leads to diminished response to new antigens. The thymus produces a hormone, the thymic humoral factor. This factor is believed to regulate lymphopoiesis in the thymus and other lymphoid tissue. Development of the human embryonic thymus begins at around 60 days after fertilization. Size and complexity of thymus gradually increases. This differentiation will only continue for a certain period of time and then stop if it does not receive a stimulus to continue growth. Immature lymphocytes begin to flock at and in the thymus of human embryos at about 90 to 100 days after fertilization. Most of the immature lymphocytes have come from the yolk sac and fetal liver rather. In humans, thymus is a bilobed structure. In birds, the lobes entirely separate into two nodular sacs, one on each side of the jugular veins. For humans and most mammals, the thymus lies at the top of the heart, at the ventral midline of the body in the thoracic cavity. The lower part of the lobes rest over the front of the heart, and top of the thymus wraps around the windpipe. Each of the two lobes of thymus are exposed in a capsule. Within each thymic lobe is a structured organization of lymphocytes and reticular cells. The lobe is divided up in a rough honeycomb appearance by membranes called trabeculae or sometimes septa, which extend from the outer capsule into the thymic lobe. Each division of the tissue by a septa is a lobule. Each lobule have an outer layer of darkly stained cells called the cortex, consisting of cells which turn out to be lymphocyte cells mixed in with many blood vessels, and an inner core called the medulla consisting of some lymphocytes but predominantly epithelial cells. Immature lymphocytes arrive in the thymus through a single artery. Similar to the bone marrow, the thymus has a closed circulatory blood system, one entry and exit point. The blood vessels have several physical layers surrounding them called the blood-thymus barrier. This barrier consists of membranes, connective tissue and cells which play an important part in restricting which cells are allowed to cross into the medulla of thymic lobules and which cells are flushed straight through the thymus. The blood-thymus barrier is known to be much weaker where the vessels pass through the medulla of thymic lobules, and cells crossing to and from the vessels can be observed in this region. Cells do not apparently cross from or to blood vessels in any other area of the thymus. Immature lymphocytes are allowed to pass from vessels into the medulla tissue, and only mature lymphocytes are allowed to pass back into the blood vessels. Once the immature lymphocytes have passed the blood thymus barrier, they are called thymocytes. Over 10% of all thymocytes are found densely packed into the cortex region of lobules, with just 10% of thymocytes found in the medulla regions. These cells are at different stages of maturation. Cortex thymocytes are the most immature, and medulla thymocytes are close to complete maturation and preparing for release back into the bloodstream. Of all the mature lymphocytes that enter the thymus, only 10% leave as fully differentiated cells. The rest die at some stage during the maturation process in the cortex or medulla of thymic lobules. Clearly, vast number of immature thymus-dependent cells must be produced by the bone marrow to ensure that enough cells leave the thymus to maintain the adult immature immune system. Adenoid tonsil right lymphatic duct entering in thymus, heart, lymph nodes, cortical epithelial cells, spleen, thymus portion, medulla, cortex, trabeculae, fibrous capsule, trabeculae, that portion extended view, cortical epithelial cell, thymocytes, trabecula, fibrous capsule, cortex, medulla, dendritic cell, macrophage, blood vessel, medullary epithelial cell, cortical epithelial cell, thymocytes, trabeculae, fibrous capsule, cortex, medulla, dendritic cell, macrophage, blood vessel, medullary epithelial cell. Location and structure of the thymus. Education of lymphocytes. Education in the sense they are training. Okay. The bone marrow produces lymphocytes that can attack wide range of antigens, including antigens of our own cell. The bone marrow makes no distinction between those lymphocytes that will benefit us and those that would harm us. Immature lymphocytes are unable to respond to any allergic changes, so their migration from bone marrow to the thymus is uneventful and of no consequence, good or bad, to us. The swarm of cells is then educated in the thymus. There is a dispute over which cells in the thymus are the instructors and educators. Some say it is the epithelial reticular cells. Others say the education comes from a population of dendritic cells found in the thymus that are possibly a subgroup of phagocyte cells produced in the bone marrow. So this portion is open for research. We need to find out education comes from either from dendritic cells or from epithelial cells. The educating cells present on antigens to the immature thymocytes. Those that are unable to respond to the presented antigen die. Those that don't respond are assumed to be protective and beneficial for us and will mature to leave the thymus. It is called negative selection also. So lymphocytes are also exposed to MHC molecules, and those lymphocytes which can recognize MHC are selected and can move out of the thymus. This type of selection is known as positive selection. There are some problems, however. The cells in the thymus do not have all our antigens available to use in educating thymocytes. Some antigens from specialized organs such as brain and other hair follicles are not present in the thymus. In summary, when thymus is the detector of T cells into a viable defense force and removes non-compliant and dangerous self-reactive immature lymphocyte precursors, then secondary lymphoid organs. While primary lymphoid organs are concerned with production and maturation of lymphoid cells, the secondary or peripheral lymphoid organs are sites where the lymphocytes localize, recognize foreign antigen, and mount response against it. These include the lymph nodes, spleen, tonsils, adenoids, appendix, and clumps of lymphoid tissue in the small intestine known as Peyer's patches. So, primary lymphoid organs are concerned with the production and maturation of lymphocytes. The secondary or peripheral lymphoid organs are sites where the lymphocyte localized, recognize foreign antigen, and mount response against it. They trap and concentrate foreign substances, and they are the main sites of production of antibodies. Some lymphoid organs are capsulated, such as lymph node and spleen, while others are non-capsulated, which include mostly mucosa-associated lymphoid tissue. Spleen in particular. The spleen is made up of B cells, T cells, macrophages, dendritic cells, natural killer cells, and red blood cells. The spleen filters antigens directly from the blood that passes through it, and migratory macrophages and dendritic cells bring antigen to the spleen via the bloodstream. An immune response is initiated when a macrophage or dendritic cell presents antigen to appropriate B or T cells. In the spleen, B cells become activated and produce large amount of antibody. The spleen has a thin connective tissue capsule from which short septa extend inward. These septa are in turn connected to a complex reticulin framework. There are two distinct components of spleen, the red pulp and the white pulp. The red pulp consists of large number of sinuses and sinusoids filled with the blood and is responsible for the filtration function of the spleen, while the white pulp consists of aggregates of lymphoid tissue and is responsible for the immunological function of the spleen. Splenic artery, splenic vein, hilum, arterioles and capillaries, splenic artery, splenic vein, central artery, red pulp, white pulp, venous sinus, splenic cords, trabeculae, capsule. Red pulp. There is a complex system of blood vessels within the red pulp arranged to facilitate removal of old or damaged red blood cells from the circulation. A small portion of the splenic blood flow passes through more rapidly without undergoing this process of filtration. White pulp. The white pulp contains T cells, B cells and accessory cells. The purpose of white pulp is to generate an immunological response to antigens within the blood. The white pulp is present in the form of periarteriolar lymphoid sheath. The sheath contains B cell follicles and T cells. At the edge of T zone is a region known as the marginal zone where larger lymphocytes and antigen presenting dendritic cells are located. Then lymph nodes. The lymphatic system panels the circulatory blood system. It is particularly guarded by lymph nodes which are found throughout the body. Composed mostly of B cells, T cells, dendritic cells and macrophages, the nodes drain fluid from most tissues. Antigens are filtered out of lymph in the lymph node, therefore returning the lymph to the circulation. In a similar fashion as the spleen, macrophages and dendritic cells capture antigens and present them to the B cells, consequently initiating our immune response. Lymph nodes are small bean-shaped structures lying along the course of lymphatics. They are aggregated in particular sites such as neck, axillae, groins, and para-aortic region. Lymph nodes have two main functions. Phagocytic cells act as filters for particulate matter and microorganisms. Antigen is presented to immune system. Lymph nodes have a fibrous capsule from which trabeculae extend towards the center. The node is made up of three components: lymphatic sinuses, blood vessels, parenchyma, cortex, paracortex, medulla. Different lymphatic vessels valve to prevent backflow: capsule, sinus, nodule, cortex, hilum. Efferent lymphatic vessel. Structure of lymph node. Inward flow lymph, fibrous covering; outward flow lymph, foreign substance filtered out of lymph and it is through the lymph node. Cortex. B cells. These enter the lymph node via high endothelial venules or high endothelial venules, HSV. If activated by antigenic stimulation, they proliferate and remain in the node. Unstimulated B cells, however, pass out rapidly from the node to return to the general circulation. Activated B cells within the lymphoid follicles are known as follicle center cells. The pale staining central area of a secondary follicle is known as germinal center, and it is surrounded by a mantle zone consisting of small native B cells and a few T cells. The follicle center cells within the germinal centers consist of cells with cleaved nuclei, centrocytes, and cells with larger, more open nuclei and several nucleoli. Centroblast. Stimulated mature B cell responding to antigen change into centrocytes and then centroblasts. The centroblasts leave the follicle and pass to the paracortex and medullary sinuses, where they become immunoblasts. The immunoblasts divide to give rise to plasma cell or memory B cell, which are ready for their next encounter with specific antigen. Accessory cells, lymphocytes alone, are not to make an effective immune response. They are assisted by so-called accessory cells. This may be grouped as follows: sinus macrophages, highly phagocytic; tingible body macrophages, ingest cellular debris in germinal centers; marginal zone macrophage, found beneath the subcapsular sinus. Then follicular dendritic cells. Then paracortex. The paracortex contains lymphocytes and accessory cells along with supporting cells, and it is the predominant site for T lymphocytes within the lymph node. T cells. The various types of T cell enter the node from the blood via the HEV. Then when activated, they form lymphoblasts which divide to produce a clone of T cells responding to specific antigen. Activated T cell then pass into the circulation to reach peripheral sites. Accessory cells. Interdigitating cells are numerous in the paracortex and they act as antigen presenting cells. Medulla. In the medulla comprise large blood vessels, medullary coats, medullary sinuses. The medullary coats are rich in plasma cells which produce antibodies that pass out of node via the efferent lymphatic. Macrophages are also numerous within the medulla. Then lymphatic system. Lymph passes into the node through the afferent lymphatic into the marginal sinus through the cortical sinus to reach the medullary sinus before leaving via the efferent lymphatic. Particulate matter in the lymph is removed by macrophages. Antigens are taken up by antigen presenting cells and this facilitates the specific immune response. Less than 10% of lymphocytes enter the node in the lymph and the large majority enter from the blood via the HSV. Lymphatic system, cervical lymph nodes, thymus, palatine tonsil, right lymphatic duct, axillary lymph nodes, spleen, cisterna chyli, thoracic duct, inguinal lymph nodes. Lymphocyte recirculation. Lymphocytes and some mononuclear phagocytes can recruit between lymphoid and non-lymphoid tissue. This helps in allowing lymphocytes to be exposed to the antigens which they recognize and is therefore valuable in the distribution of effector cells of the immune response to the sites where they are needed. Lymphoid tissue containing lymphocytes and macrophages, capsule, lymph node, blood vessels, heart, capillary blood, intestinal fluid, cell products and debris, pathogens, lymph vessels carrying lymph, lymphatic system, route of the flow of lymph in body. The recirculation is a complex process depending on interactions between the cells of the immune response and other cell types such as endothelial cells. Virgin lymphocytes move from the primary to secondary lymphoid tissues via the blood. Activated lymphocytes move from the spleen, lymph nodes and malt into the blood and hence to other lymphoid and non-lymphoid tissues. Antigen presenting cells such as macrophages and dendritic cells may carry antigen back to lymphoid tissue from the periphery. The complex pattern of recirculation depends on the site of activation of the lymphocytes, the adhesion molecule expressed by endothelial cells and presence of chemotactic molecules which selectively attract particular populations of lymphocyte or macrophages. Mucosa associated lymphoid tissue. In addition to the lymphoid tissue concentrated within the lymph node and spleen, lymphoid tissue is also found at other sites, most notably the gastrointestinal tract, respiratory tract, and urinogenital tract. Gut associated lymphoid tissue. It comprises tonsils, adenoids, Walde's ring, Peyer's patches, lymphoid aggregates in the appendix and large intestine, lymphoid tissue accumulating at age in the stomach, small lymphoid aggregates in the esophagus, diffusely distributed lymphoid tissue, lymphoid cells and plasma cells in the lamina propria of the gut. Large aggregates of GALT have distinct B cell follicles and T cell areas. Antigen presenting accessory cells are also present. And Peyer's patches. These are quite large aggregates of lymphoid tissue found in the small intestine. The overlying dome epithelium contains large number of intraepithelial lymphocytes. Some of the epithelial cells have complex microfolds in their surfaces. They are known as M cells and are believed to be important in the transfer of antigen from the gut lumen to Peyer's patches. Peyer's patches facilitate the generation of the immune response within the mucosa. B cell precursors and memory cells are stimulated by antigen in Peyer's patches. Cells pass to the mesenteric lymph nodes where the immune response is amplified. Activated lymphocytes pass into the bloodstream via the thoracic duct. These cells then home in the gut and carry out their final effective function. HSVs are not present in Peyer's patches and the mechanism by which cells home on the mucosal sites is unknown. Now let's have a quick summary. Immune system in vertebrates is very complex and made up of number of organs and cell types. The various cell types are generated by the presence of hematopoiesis in the adult bone marrow. Immune organ system is divided into two: primary lymphoid organs and peripheral or secondary lymphoid organs. Primary immune organs are bone marrow and thymus. Peripheral immune organs include spleen, lymph node, and Peyer's patches.